In this video, we're going to continue our discussion of specific immune responses. In particular, when looking at that third line of defense, we're going to be concentrating here on the cellular immune responses, and the next video will be on the antibody-mediated responses.
To start with, let's define that when we're talking about specific immunity, the main players are going to be our lymphocytes. And we talk about two different lymphocytes, B cells and T cells. B cells get the term B from the term Bursa fabricius, and that's just because it was first discovered in avian or bird. T cells get the T from the word thymus because they're thymic derived. So B cells, they're going to be from the bone marrow. They stay in the bone marrow and they mature in the bone marrow and they develop into what we refer to as plasma cells, which are going to be antibody secreting cells. T cells, they're going to be derived from the bone marrow, but then they go to the thymus and that's where they mature, and hence we call them thymic derived or T cells. So when we look at the specific response, there's two types, cellular immunity and humoral immunity. Let's talk a little bit about cellular immunity. When we talk about cellular immunity, it means that it's going to be cell mediated, and what we're referring to there is the T cell ability. We're going to learn here a little bit in this review about the different types of T cells. The different types of T cells get activated by that type of antigen. So then it determines which type of response we're going to provide. So it's mediated, the response is mediated by the T lymphocytes, so it's cell-mediated or cellular immunity. And again, it's going to be associated specifically with the type of antigen. And so our T cells are going to be numerous, and they are going to be about 70-80% of those lymphocytes that are circulating in the blood. So let's talk a little bit about T cells. Again, we mentioned that they were formed in the bone marrow, but then they go to the thymus, and that is then where they mature. They mature in the thymus under the influence of a hormone known as thymosin. So when the T cells are going through that maturation process, they're going to develop and they're going to differentiate either into what we call a CD4 or T4 cell, or into a CD8 or a T8 cell. The CD cluster differentiation is a marker, is another one of those cell markers that we referred to in review 1 for part 2. So again, we have T cells. It's going to be the most numerous of the circulating lymphocytes. They're going to differentiate, mature and differentiate in the thymus, and then they're going to move back out to the blood as either a T4, which has a CD4 marker on it, or a T8, which has a CD8 marker on it. So, looking at the T cells, a couple things that you need to understand is, again, we have the two different types of T cells, but on each T cell, there's a piece known as the T receptor, or the TCR. That TCR has two components, okay? One is the antigen receptor. That's going to be the piece that recognizes the epitope of the antigen. So if you'll recall in part 1, we talked about the fact that an APC, an antigen presenting cell, is going to then take and put a piece of antigen on its surface using an MHC marker. So the antigen receptor is going to recognize the epitope, okay, that is on that APC. The CD marker is either going to be CD4 or CD8, okay, and this is actually going to recognize that MHC component on the APC. So the APC, as you will recall, had two pieces. It had an MHC with a piece of antigen, okay, or the epitope. So it had two pieces in the presentation. The T cell has a two-piece receptor marker, if you will, okay, and showing down here. So we have two different types. We have a T helper and a T cytotoxic. T cytotoxic here has the CD8, so it's also known as a T8 cell. T helper cells have a CD4 marker, so we refer to it sometimes as the T4 cell. Okay, so two different types of T cells. Both of these are part of the cell-mediated response, okay? And because we have two different types of T cells, we're going to have different responses to different antigen. So I want to take us back just a moment. You will recall that in part 1 of Unit 4 and even part 1 of Unit 2 review, okay, or excuse me, part 1 of part 2 Unit 4 review, we talked about some different terms. So I'm going to go through and talk about intracellular antigen versus extracellular antigens. Okay, so intracellular antigens, remember, this is going to be a virus or it's going to be cancer, along those lines. Extracellulars could be toxins, bacteria, etc. Okay, let's just, for ease sake, say intracellulars, we're going to represent that by being a viral infection. Extracellular, we're going to represent that by being a bacterial infection. Okay, so if this is the tissue, so it's, let's just say it's your respiratory tissue. Okay, so nucleated cells, so they have the ability to present or to be invaded by viruses or things like that. Okay, so if we're looking here and we look at the virus, okay, so if it's a viral infection, it's going to come in, it's going to come into the body, and remember, viruses are intracellular, so I'm going to put it as the letter V. They have to come in and they have to get inside our nucleated cells here, and then they're going to take over our cells, use our cells to replicate, divide, and then they will come out of the cell, infect other cells, etc. But while they're doing their damage and stuff, notice where they are. They're inside the cell. Okay, whereas over here, here's the nucleated cells. For most bacteria, when they come in, they stay here. So we're going to represent them with a B. Okay, and notice they stay outside of the cell. Because then what they can do is they can release, if you recall back, they can release various toxins, enzymes, or chemicals, and those things can come in and do destruction or damage, okay? But again, they stay out. So if you are the body and you get a virus, or if you get a bacterial infection, can you fight them the same way? No, and you know this just intuitively. If you get a bacterial infection, what's the first thing you do? You go to the doctor if you got a strep throat or something, and you go and you ask for an antibiotic. Okay, the antibiotic, why? Because it's going to be able to recognize the bacteria and clear it. If you get a cold, do you immediately go to the doctor? No, you stay home and you get chicken soup. Why? Because antibiotics for the majority of viruses are non-effective. Okay, so here's why. So notice here in the intracellular, the virus is inside the cell. If we're going to get to the virus, inactivate it, to destroy it, to prevent it from doing further damage, what do we have to do? The body's immune response is going to have to come in, it's going to have to find the infected tissue, and it's going to do that through that inflammatory response. So because the virus entered into the cell immediately knows, hey, I've been infected, and it's sending out chemical signals. But your body's going to have to scan these cells, and it's going to be looking. For instance, number 1 here, it's got a virus infection. We need to make sure we're doing something to cure that. Number 2 here also has the virus. But number 3, notice it doesn't have. The virus hasn't spread to it. So in order for the body to get rid of the virus, we want it to do something that can take out this cell and this cell, but not that cell, because that cell has not been infected and is still a good, healthy cell. So our immune response to an intracellular is we're going to want it to go in and we're going to want it to target infected cells. Okay, and when it targets the infected cells, it's going to have to kill the cell to get rid of the virus, but it's going to have to be controlled. We can't just have it as an uncontrolled, hey, let's go kill every cell, because we can't do damage to surrounding cells that are non-infected or still healthy. Okay, let's compare that then to the extracellular response. So notice again here, if your body is coming in and it's bringing immune responses to the area, and it will, because the bacteria are going to be releasing enzymes, toxins, different things that are doing damage, so the body's undergoing damage. It's sending out chemical signals. It's calling in those immune cells to the area to start eliciting a response. But when it comes here and it's scanning, it's going to be able to see this bacteria because it's outside of the cell. So it's not hidden inside the cell. So it's going to be able to come in, and it's going to be able to, your immune responses, you're going to want it to attack the bacteria directly and any of the toxins and stuff, but you don't want it to have any impact on these cells because these cells are still good and healthy. So in this case, we're going to actually target the bacteria, the invader, the antigen. We're going to kill the antigen or deactivate it, depending on if it's a toxin or whatever, and we're going to do no harm to self cells. So you can see there's two different distinct responses that we're going to elicit. And we're going to do these based on the T cell mechanism. So how many types of T cells do we have? 2, CD4 and CD8. How many types of responses or antigens do we have? Generically, and the widest capacity of classification, we have 2 types of antigens. We have intracellular and extracellular. So guess what? We have 1 T cell for each type of response. The T cell that you activate is going to be dependent on the type of antigen that enters the body. So we have a helper T cell and we have a cytotoxic T cell. Helper T cells are going to do just what the names imply, okay? They're going to come in back to here. If we're attacking the bacteria itself in an extracellular, we're going to do that because we're going to make antibodies. Again, remember we talked about you go to the doctor to get antibiotics. So if we're going to make antibodies, B cells make antibodies, and the T4 cells are going to activate the B cells. So in the case of an extracellular response, we're going to have T4 cells, which are also known as helper cells. They're going to activate B cells. They're going to then elicit antibodies, and we're going to be able to get rid of bacteria. In the case of the intracellular, okay, and I'm going to go through these slides. I'm flipping through real quick here, slower in just a second. We're going to elicit cytotoxic cells, which are CD8. So here we're going to be doing cytotoxic, which are the T8, and as the name implies, cytotoxic. They're toxic to cells. Which cells? Our cells. So in the case of an intracellular, we're going to activate the T8 cells, and we're going to have a cytotoxic response. Okay, so that's kind of our overview. T helper cells, just to give you some more specifics about it, these are the ones that have the CD4 marker, so we also refer to them as a T4 cell. Okay, they're going to be used for the extracellular antigen. Okay, and in particular, we're going to be using the T helper 2 because they're going to help activate the B cells to differentiate, and then B cells are going to produce antibodies, and AB just stands for antibodies. Okay, so helper T's, there's 2 types. There's a T helper 1, which is going to be used in a lot of different areas. They're going to help with activating T cells, macrophages, delayed types of hypersensitivities, and then helper 2s are going to be for that B cell differentiation, antibody production, and in particular extracellular antigens. But as the name implies, they're helper cells. They're helping to elicit other or activate other immune cells to elicit an immune response. Okay. Big thing to notice is that they're going to be activated using the APC that has a class 2 MHC. Cytotoxic T cells. These, if you'll recall, these are the intracellular. They're going to be elicited during an intracellular antigenic response. These are referred, they have the CD8 marker, so we refer to them as T8 cells. And unlike T helpers, which have subsets, the cytotoxic do not. It's just a CD8 cytotoxic T cells, and they're going to go in and they're going to attack and destroy cells that are infected with an internal antigen, okay, an intracellular antigen. And notice they get activated by MHC class 1. So One of the things that I like to tell students of how they're like, I can't ever keep apart CD4, CD8, MHC1, MHC2. Here's my quick tip, if you will. Okay, MHC class 1 with a CD8, and you have MHC class 2 with a CD4. 8 times 1 equals 8. 4 times 2 equals 8. That's how I keep them a part of my head. It may not work for you, but when I was trying to learn this for the first time, I had to have little catches and things to help me remember. So MHC class 1 are for the intracellular CD8 T cell response. MHC class 2 are for the extracellular bacterial cells CD4 response. So how do we activate the T lymphocytes? Each one of these is a very long process. There is a video on each one, as well as a handout that's a whole one-page handout in your lecture notes. I'm going to refer you to those, but a couple things I want you to pay attention when you're looking at those two videos is understand first that the process starts because the APC comes into the site of infection. It will engulf that antigen. It's going to display that antigen on the surface using an MHC or an MHC class 2. Again, there's videos on APCs, and it's part of part 1 review for the unit 4, part 2. Okay, so this is all that APC presentation. Which type of antigen will determine which MHC is displayed? If it's an internal intracellular viral type antigen, then the APC is going to display it with MHC1. If it's a bacterial external extracellular antigen, then the APC is going to use an MHC class 2. This then dictates which T cell, which population of T cell is going to then be activated. If it is an MHC class 1, then it's going to be using the CD8 because it is an intracellular response and we got to have the cytotoxic cells. If it is an MHC, if it's an extracellular antigen like a bacteria, then it's going to use an MHC2. That's going to activate our CD4 cells, which are going to be those that then lead, they're the helper T cells, which are ultimately going to lead to B cell activation and antibody production so that we can clear the bacteria. So it is that matching of MHC with CD4, which is all dictated by the specific antigen, which then helps us to elicit the proper immune response. Hence the reason we say it is specific immunity, because the specific antigen will elicit the specific response. Is it going to be B cell antibody production, or is it going to be intracellular and cytotoxicity?
Our third video is going to look at that last piece of specific immunity, looking at the antibody-mediated responses. So don't forget to look for that one, and also go back in and look for that T cell activation video.